we use a lot of the same methods as traditional machine learning, so it's not a big difference. All right, so I guess we could start now. Eric, what do you think?
Yes, please. Yeah.
All right. So okay, so by the way, Shia, you can take your time. I mean, we typically stop at 1, but if you have some more time for questions or whatever, it's really fine. And you can make it informal. It's a seminar, but we're really open to discussion, and so okay. So it is my great pleasure to introduce the professor of Dr. Xia Ning on deep generative models for molecule optimization. So Dr. Ning is an associate professor in the Biomedical Informatics department and the Computer Science and Engineering department at the Ohio State University. She received her PhD in computer science in engineering from the University of Minnesota, Twin Cities, in 2012. And I have to say that I met Xia over there while I was doing my postdoc in the same lab. Specific applications of her work, she's going to talk more about it today, include new molecule generation and drug candidate prioritization for drug discovery, cancer drug selection for precision medicine, and information retrieval for electronic medical records. And she won the 10 years highest impact paper award in 2020 at the IEEE International Conference on Data Mining. So without further ado, I let Xia present her talk today to you.
Thank you, Christian, and thank you for having me here. It's my great pleasure to be here. So probably I'll be talking about our very recent work on deep generative models for molecule optimization. So as the title suggests, it's about deep learning, it's about generative models. And in terms of the applications, it's about molecules, it's about drugs, it's like applications in chemistry. So before that, I really want to present this top 9 takeaways. So Stanford published a AI index report last year. So in that report, they analyzed the current trend of AI's issues and future directions, and they listed these top 9 takeaways. So these top 9 things probably are the most representative current AI trends and future directions. So among those 9 things, actually, the top 1 thing is about AI investment in drug design and discovery, and it has been increased significantly since last few years. And by the way, this is about the AI index primarily in the US, but I think very similar things could, very similar conclusions could also apply for Canada. So the first one is on AI for drug discovery, and the third one is about generative models. So Christian must be very familiar with generative models for computer vision for images. I mean, that is typically the generative models are used. But here in our case, we want to develop some generative models for a very different application, that is drug design and drug discovery. And other interesting items along the list includes like ethics and surveillance issues. So it's a very nice report. So I do suggest if you can take a look. It's a very nice report, very in-depth report. So here is a very quick line of my talk today, some introductions and our work on deep generative models. So I want to also give you a quick introduction about the drug design, because this is not a very typical application area where computer scientists used to work on. So for drug discovery or drug design, so this is more or less a very typical process. So on the left-hand side of this figure, you see the traditional drug development process. So drug discovery or drug development starts from the therapeutic target. So that is, we have a disease of interest, and for that disease, we can identify the therapeutic target. Typically, it's a protein. So that protein is the reason, or the genes expressed into that protein is the reason for that particular disease. And after we identify the therapeutic target, we want to develop some potential drug candidates. So oftentimes this candidate development starts from a very high, large-scale screening of many molecules. Like typically we have millions of molecules and we do a very quick, high-throughput screening. It could be through some wet lab or bench work, or we can also do the virtual screening, like we use AI methods or machine learning method to predict which compound can have some potentials as a drug against this disease. So after the large-scale high-throughput screening, we can identify oftentimes 100, 200 potential candidates, and those candidates will go through downstream analysis and testing. So oftentimes we will have some lab tests or some secondary or confirmatory screenings to better test the properties of those molecules. And out of there, oftentimes we'll get 20 or 50 compounds, which will go through the following cell culture assays, and hopefully 10 or 20 of them can go through the small animal tests. And after that, oftentimes we can identify 1 or 2 candidates which will go through the human clinical trials. So for human clinical trials, oftentimes typically we have 3 phases. In the first phase, we test the safety or toxicity of that drug candidate on human beings, on those volunteers. And so this is for safety issues. So we want to make sure that the future drugs are safe. They do not have severe side effects. They are not toxic. Then in the phase 2, we tested the efficacy of those drugs, or whether those drugs can treat the disease in an efficient way. And after that, it's phase 3. So in phase 3, we tested the efficacy and drug safety again, but on a large scale, on a larger population or a larger group of subjects. And hopefully, in the end, 1 drug candidate can survive all the clinical trials and can be approved by FDA here or by the Health Canada in Canada. So this is a very typical, very traditional drug development process, and this process can take more than a decade and billions of dollars. So it's very expensive, very time consuming, but the successful rate is super, super, super low. So oftentimes people spend like 10, 20 years without nothing out of the entire pipeline. So that's where we see the challenges and opportunities for data-driven methods, for AI methods. If we can speed up the process, if we can make the process more accurate, then we can have a huge impact on the entire process and in the entire healthcare system. And recently, there is a new paradigm for drug discovery, something called drug repositioning. So the idea is we want to use some FDA or Health Canada approved drugs, but we use those drugs for different disease. For example, some drugs are developed for, let's say, SARS. They can be used for COVID treatment. So that's something called repositioning. Reposition an existing drug for a new therapeutic indication or for a new disease. So the good thing about drug repositioning is that we don't need to go through all the animal models, all the assays, and also the phase 1 toxicity test, because these are FDA approved drugs. So they have gone through all these tests. So they are not, they are not toxic for sure. So we just need to test whether these drugs can treat the new disease. So in phase 2 and phase 3. So for drug repositioning, as you can tell, it's going to be much faster, much more efficient, and we can save a lot of time, a lot of effort, and a lot of course, money if we can have a very promising reversible drug, and we just need to do phase 2 and phase 3. So hopefully this can give you a very quick idea about the drug discovery process. And what we are doing on drug optimization or molecule optimization actually happens somewhere here. in the very early phase of drug discovery, in the design and the synthesis phase. So the idea is after we do some screening, we identify some molecules. Those molecules are promising, but not ideal. They may have some desired properties, but they may have some other undesired properties, or their properties are not good enough. So we want to change the structures of those molecules so as to actually modify their properties, oftentimes to improve some desired properties and to remove some undesired properties. So we do this type of optimization, and oftentimes we just want to change the structures of those molecules a little bit, not the entire structure, but some structures of these molecules in order to change their properties. And we want to develop AI method to automate this process rather than having all the chemists to do the work manually. So that's the idea behind our work. But from the entire drug discovery process, you can tell there are many challenging AI problems. For example, how to identify the drug candidate, how to find the compounds that are most drug-like, how to predict drug properties like activity, efficacy, toxicity, and other properties that a successful drug needs to have. And selecting the right drug to the right patient. So this is the key in precision medicines. Not everyone responds to a same drug in the same way. So we want to give the best treatment to each individual, personalized to each individual. So that's the drug selection problem. And many drugs will have some adverse drug effects, and oftentimes those effects cannot be observed during the clinical trials, simply because the clinical trial is really a small-scale trial. But after the drug is used among a huge population, oftentimes we'll see something unexpected or never known before. So we want to predict whether drugs, and in particular, many drugs together, can induce some adverse drug effects. And this is for pharmacovigilance purposes. And sometimes FDA or Health Canada will recall those drugs with very bad adverse effects. So that's a very actually bad thing for drug discovery and also for the public health. And therapeutic indication prediction. So in order to use an existing drug for a new disease, we need to have a signal or have some kind of knowledge or evidence showing that it's possible. So that leads to a problem to predict whether the disease can be treated by another drug, or whether this a drug can be used to treat a different disease. So there will be the therapeutic indication prediction problem. And of course, the molecule optimization and the generation design. So that's what I'm going to talk about today. And actually, there is another very interesting problem. It's about the chemical retrosynthesis. So let's say we have a molecule, we think we have a structure of the molecule in mind, and we think this is a good molecule. And in the end, we need to make this molecule. We need to synthesize this molecule. And to identify the synthesis path of that molecule is not a trivial thing. And even for very experienced chemists, they need to have a lot of knowledge, a lot of prior experience in order to design a very efficient synthesis path in order to identify the molecules that can be used to synthesize the targeted molecule. So we have been also working on the retrosynthesis path planning using AI. But today I'll talk about our compound optimization through generative models. So again, some more background about molecule optimization. So this is a critical step in drug discovery to change the to improve the desired properties of drug candidate through chemical modification, through changing the small fragments or small substructures of the chemical molecular graphs in order to change or modify the properties. So this is typically done by fragment-based screening or synthesis. So fragment-based screening is, let's say we have a molecule in mind, we want to change that molecule a little bit, and then we will look for similar molecules, similar in the sense of their molecular graphs. We can use graph similarity-based method to identify similar molecules, and we do all the wet lab experiments to test those molecules. So those molecules are similar to our initial molecule, but they are different in certain fragments. So that's the fragment-based screening. Or we do synthesis, like we want to change a fragment, then we use synthesis reactions in order to break a bond or attach a new fragment there. So this is what is typically done in drug discovery. So very recently, people started to look at this problem using AI. I mean, as you can imagine, AI is everywhere and can be used everywhere. So starting from 2018, people start to use AI method, deep learning method, to help the optimization or generation of new molecules. And oftentimes people, and it's as you can imagine, it's based on either representation learning over molecular graphs or representation learning over sequence representation of molecules. Actually, each molecule can be represented by its formula, and that's a sequence, and people can use natural language processing methods to analyze the sequence. And from there, people can generate new molecule structures and with better properties. And oftentimes it's going to be an encoding, decoding type of framework, or reinforcement learning is also popular method here. So some known issues about this type of deep learning method is oftentimes many methods just generate a new molecule structure from scratch, and they cannot really guarantee that the new structure is similar to the initial molecule structure, or they typically focus on one particular property to optimize. So it's not a very general framework for all or for many desired properties. So we want to tackle these two issues in our work. So some very key ideas before I show our framework. So if you think about how to optimize a